Here's some questions that you guys hopefully should know the answer to from a while back. So, I don't know, maybe you forgot. But what are the sections of DNA called? Like the things that you wear on your legs? Genes. Okay. Spelled with a G. G-E-N-E-S, not J-E-A-N-S. Okay, so it's a little bit different, but I saw people every year that spell that wrong. Alright? So, what does DNA make? Like, what's the point of DNA? At the end of these processes, we're going to get some stuff called, find it in nuts and meat and proteins. Good, Jeremiah. Proteins. So, DNA makes proteins. Okay? And what are the building blocks of those proteins? This is from a few months ago. Amino acids. Does that sound familiar? Amino acids? Alright, transcription and translation. Okay? We're going to look at the process that we have to go through to get from the DNA in our code to the proteins that help to make us who we are and help with body functions and things like that. So, I think I kind of previewed already, but why are proteins important? Because some proteins actually become structures in your body, like muscle tissue. So, like I've said in the past, I'll say it again, if you're a lifter and you do your protein shakes, it's because it's going to help your body to recover from the lifting. When you lift weights, you're actually tearing your muscle a little bit. Okay, and so when your muscle repairs itself, that's how you build up your muscle. Okay? So by drinking protein shakes, you're giving your body the things that it needs in order to build that muscle back up. Okay? They're also good for becoming enzymes that help control chemical reactions that happen in your body to help break things down, put things together, that kind of stuff, chemical processes. Okay? Alright, so now we're going to get into what the heck we did yesterday with the Legos. You ready? There are 3 types of RNA. Anybody thinking they know the connection now? 3 types of RNA. There's mRNA, rRNA, and tRNA. Okay? Anybody have an aha? Yes? There's 3 types of RNA. What did we do yesterday with the Legos?
There was the builder, there was the flyer, and here was the instruction or director.
Okay. So, the 3 types of RNA were represented by you guys. Like yesterday in your Lego group, there were 3 people in your group that were building the Lego car. So you guys were representing the 3 types of RNA. Okay? So as we go through, I'm going to explain what each of these things does, and you'll see how it relates to what we did yesterday in our activity. Okay?
Did people build helicopters too?
Very end of the day. Because I didn't have enough of my cars. So, too many people. They didn't get to finish them. They were too complicated. Alright. mRNA is messenger RNA, brings instructions from the DNA, which is in the nucleus, and takes them out into the cytoplasm to the ribosome, which is kind of like the assembly line that puts the protein together. So the mRNA, which one of you guys in your groups was an mRNA?
E-man.
What was the role? To do the instructions. So the person, so this would be like the director of operations, the person who read the instructions in the activity yesterday, you were being the mRNA. You had the instructions for how the protein got put together. Okay, or how the car is representing our protein. Okay. So the rRNA is ribosomal RNA, which makes up the ribosome. Okay, the ribosome binds to the mRNA and takes amino acids and puts them into the right order. So which person in the group, what role was representing the rRNA? The builder, yeah. So putting them together in the right order. So the rRNA was represented by the builder. Okay, the car that you guys were building is representative of the protein that gets built by these 3 types of RNA. You guys with me? How am I doing staying under your Am I doing okay? I'm trying. Okay. Alright, the last type is tRNA, which is called transfer RNA. And obviously, it acts as the supplier. The tRNA will take amino acids that are floating around in the cell and will bring it to the ribosome so that they can be put together into a protein. Right. So that represented the Lego race supplier. So the Lego pieces themselves are representative of amino acids. So we take amino acids, put them together to make a protein, use the Lego pieces and put them together to make the car. Does everybody understand the analogy that I made? Okay, and I think, I don't know, it's a complicated process, but for whatever reason, I think that analogy really helps, and people tend to do really well on this test because of it. I don't know why, but people tend to do pretty well on that. All right, so remember DNA replication from earlier this week and how DNA replication makes 2 double-stranded DNA molecules? You guys remember that? I'm going to tell you this for a reason because now I'm going to contrast what we're about to do with that. So keep that in mind. All right, so here's the picture of it. So how do we describe DNA that's replicated? It's kind of a longer word. Begins with an S.
D-O, no.
Semi-conservative, right? It's semi-conservative because we keep half of the old DNA, in this picture represented by white, and then we add on the new DNA, in this picture represented by black, so each piece of DNA is half old, half new. So it's semi-conservative because we're kind of keeping half of it. Okay? So the first part of the process of making a protein is called transcription. So this is how we get the DNA message into a form of RNA called mRNA. So the instructions. Okay? It's a process that's very similar to replication, but instead of 2 DNA molecules, okay, that are double-stranded, we end up with 1 single-stranded RNA molecule. Okay? So it's kind of the same way. There's unzipping involved, there's base pairing involved, but instead we just have 1 single-stranded RNA that comes out of it. And so that would look something like this. I think all the pictures are on your paper, which is why the packet looks so big. So this is the DNA, okay? And the DNA, see how it's unzipped? It came apart here in the middle. Now, we know it's DNA because it has what letter?
A, G, T, C.
It has the T's in it, right? Yeah. DNA has T's. But what's this one have on it?
U.
U's. So that's our RNA that's being made. So this will open up, and these letters on the DNA act as like the template. So what it'll do is say, okay, there's a C here, so the first one on their RNA is G. A is U, T is A, G is C, and we create this messenger RNA, and once it's done, it's going to separate from the DNA, and the DNA will go back together. Okay? It's a little bit more complicated than that, so let me just explain a couple of the details. There's things called exons and things called introns. Okay? Exons are segments of gene that carry code. So any of the information that gets expressed in the form of proteins is on the form of an exon. Whereas introns are the non-coding parts of genes. We have a lot of DNA in our body that doesn't do anything. It's just called junk DNA. In fact, it's the stuff that makes us unique from each other. Because think about it. Like, for example, we all have red blood cells and we all have hemoglobin on our red blood cells. Hemoglobin is the protein that helps us to be able to carry oxygen on our red blood cells so that they can get to the other cells in our body. Okay? But we all have that, right? So if we looked at that code for hemoglobin, is it going to be different among us? No, like it would be hard if we looked at that part of our DNA, it would be hard to tell us apart because we all have that. But if you look at our junk DNA that doesn't really code for stuff, that actually is highly variable, and that is a lot of times what they use to do like parental testing and that kind of stuff to see who the baby daddy is and all that kind of stuff, right? Okay. That's what we're gonna look at how that you can do that. How you can make a DNA fingerprint to do that. I want to try and do that before the Ohio State students come because we're gonna do something similar with them. Except it's a crime scene, not parental testing. Alright, so here's a picture of a chromosome, and then it's unwound. Remember, the chromosome is just made up of DNA. And a section of DNA is called a gene. And here we have exons and introns kind of alternating. So sections that are coding the exons and sections that are non-coding, which are the introns. Okay? When we first make the mRNA, both the sections are copied. The ones that do the code and the ones that don't. But before the mRNA can leave the nucleus, before it's considered to be finished, we have to take out the parts that don't code. So we're going to remove the introns. Okay? So that looks something like this. So at the top you have your DNA with exons and introns. We copy what we need, so the mRNA. So now we've turned it into RNA. But before it's completely done, we take out the sections that are non-coding, so these striped sections. So all that we end up left with for our mRNA are just the sections that will code for the protein that we need. Kind of good? Okay? Okay. Alright, so the things that cut that out are called enzymes, which, it's kind of funny, those are actually proteins in and of themselves. So you need proteins to make proteins. Okay? Which came first? No? It's complicated. The mRNA, after it's done, is going to leave the nucleus and go to the cytoplasm, to the ribosome. Alright, so all of this stuff called transcription that we just went over, all of that happens within the nucleus of the cell. Okay? Here's the picture of it just so you guys understand. This is the whole cell. This is everything we're going to talk about today. But what we just talked about all happened in the nucleus. So here's the nucleus, the gray area. So you take the DNA, you make the RNA, cut out the introns so you get mature mRNA, and then it'll leave and go out to the cytoplasm for the next step. Okay? So you have a quick check now on the top of your paper. So I want you guys to take just 2 minutes, okay, and do the quick check on your own. It's just reviewing what we just talked about, and then we'll go over it together and move into the next section.